Today, we're diving into some really incredible research published in Nature Communications. It's a story that feels a bit like a scientific detective novel. It's about a hidden biological switch and the amazing potential of using existing drugs for completely new purposes. I mean, just think about that for a second. What if the solution to a complex, heartbreaking problem was already out there, just hiding in plain sight? Well, this study explores that exact possibility for Duchenne muscular dystrophy, which is one of the most severe genetic disorders out there. So to really get why this is such a breakthrough, we have to start with the core of the problem itself. We'll call it the dystrophin dilemma. Okay, so Duchenne muscular dystrophy, or DMD for short, is the most common inherited muscle disease, and the science is super clear on this. The whole thing comes down to the complete absence of one single critical protein, and that protein is called dystrophin. So what does this dystrophin protein actually do? Well, the best way to think about it is like a molecular shock absorber for your muscles. Its main job is to literally connect the internal skeleton of a muscle fiber to the support structure on the outside. This link is absolutely crucial. It's what gives our muscles their strength and protects them from getting damaged every time they contract. And this table really hammers home what happens when you lose that shock absorber. In a healthy muscle, you've got dystrophin, and the cell membrane, which is called the sarcolemma, is strong and stable. But in DMD, without dystrophin, that same membrane becomes incredibly fragile. It's just prone to injury. The end result is this awful, devastating cycle of damage, inflammation, and muscles slowly being replaced by fat and scar tissue. But this is where the story starts to get really hopeful, because it turns out our bodies might actually have a backup plan built right in. So let me introduce you to utrophin A. The research paper calls it the autosomal homologue of dystrophin. Now that's a bit of a mouthful, but it's just a scientific way of saying it's a very, very similar protein that can do almost the exact same job as dystrophin. And the key thing here is it's made by a totally different gene, so its production is completely fine in people with DMD. And that leads to the million-dollar question, right? If the body can't make dystrophin, could we somehow convince it to just make more of its natural backup, utrophin A, to fill that gap? This is the exact strategy these scientists decided to investigate. So how on earth do you turn up the production of just one specific protein? Well, the researchers decided to target this really elegant biological mechanism. Think of it like a hidden production switch. Now, this is where it gets super cool. See, most protein production happens through a standard process, kind of like going through the front door. But the genetic instructions for utrophin A have a special feature called an internal ribosome entry site, or IRES. Think of it like a secret side entrance. This allows utrophin A to be made even when the main front door is shut down, which happens a lot when a cell is under stress. So how do you open this secret entrance? Well, the research shows it's a pretty clear step-by-step process. First, you have the IRS. That's the lock. Then there's a specific protein called EEF1A2, and that's the key. When that EEF1A2 key finds the IRS lock and binds to it, it unlocks the secret entrance, and that gives the signal to start making more utrophin A. Of course, a theory is just a theory until you test it. So the scientists first had to prove this whole idea even worked. They did what's called a proof of concept experiment, where they basically flooded mouse muscles with that key protein, EEF1A2. And the result? A nearly twofold increase in utrophin A. That was the green light. It confirmed that this pathway was a totally legitimate target for a new therapy. Okay, so the pathway works, that's great. But you can't just go around artificially boosting proteins in people. That's not a practical treatment. The real challenge, the real hunt, was to find a drug, something you could actually put in a pill that could act like that key. And so the hunt began. The team set up what's called a high-throughput screen, which is just a fancy way of saying they could test a whole lot of compounds really, really fast. And they tested a library of 262 different drugs, all of them already FDA approved for other things. And out of those hundreds of drugs, two major candidates popped out. First was pravastatin, which is a really common cholesterol-lowering drug, and the other was betaxolol, a beta blocker that's often used for high blood pressure. Both of them were found to significantly boost the levels of both the EF1A2 key and the utrophin A protein. Now, why is this so incredibly exciting? Well, because previous research had already set the benchmark. It had already shown that just a twofold increase in utrophin A is enough to make a real significant improvement in animal models of DMD. So this screen had found existing drugs that could hit that magic number. So the next step was the ultimate test. They had to see if these drugs worked not just in cells in a petri dish, but in a living animal that was actually suffering from the disease. This was really the moment of truth for the entire project. So what they did was they took dystrophic mice. This is the MDX model, which naturally doesn't have dystrophin, just like humans with DMD. And they treated them for 4 weeks with either pravastatin or betaxolol. The goal was simple: see if these drugs could deliver real, measurable benefits. And the results were, well, they were just powerful. This chart shows the change in forelimb grip strength. You can see that compared to the untreated mice, both drugs caused a clear improvement. In fact, the paper actually describes the effect of pravastatin as a striking amelioration, meaning it rescued muscle strength to a level that was almost the same as healthy mice. And the benefits went way deeper than just grip strength. When they looked at the muscles under a microscope, they saw a nearly 3-fold reduction in membrane damage. There were fewer signs of that constant cycle of damage and repair that's so typical of DMD. And the muscle fibers themselves were literally stronger and more resistant to injury. You know, the authors themselves really summed it up perfectly in their conclusion. Their work reveals that this hidden switch, this EF1A2 protein, is a truly relevant target for DMD treatments. And it shows that it's absolutely feasible to use repurposed, FDA-approved drugs to flip that switch. And that just brings us right back to that incredible idea we started with. This research began with a deep, fundamental understanding of how a single protein works, and it ended by identifying common, existing drugs that could potentially fight a devastating disease. It really leaves you with a huge question to think about. How many other cures are already out there, just hiding in plain sight, waiting for us to find them?